Today it is my pleasure to host Javier Gallego, which will be talking about the interesting topic. I think it's interesting, but also not only for plants, but also for humans. It's important to know about epigenetics because that also contributes to how we respond to the environment. And the plants is even more important. So as I said, it's my pleasure to host Javier Gallego. Just a brief introduction of the CV. So he made the thesis in IBMCP in Valencia under the supervision of Dr. David Algarí and Miguel Nazquez. He worked at that time with hormone signaling and the idea of light response. And after that, he went as a postdoc to the lab of Joan Corey in Salk Institute to continue this work on light signaling. It was a short postdoc of 1 year, and then he made a nice move to the lab of Steve Jacobsen, who is the best, I would say, PI working in epigenetics in plants. And he spent then 6 years with a lot of success with different projects all related to epigenetics. After that, he managed to come back to Spain, to the IBMCP, with a harmonica Hall petition, which was for 5 years. And during that time, he spent a lot of effort to develop his own group. And successfully, last year, he got a permanent position as a científico titular in FSIC, in the same research center. And he will talk today about the nice work doing in his group with epigenetics in plants, particularly in arachnosis. Thanks, Javier, for hosting this invitation, and I hope you enjoy your stay here.
Thank you so much. Well, hi everyone. Well, first of all, thanks for the invitation and for the kind introduction. It's a great pleasure to be here and tell you a little bit what we do in our lab. So before starting, I prepared sort of like a summary of my career, but he already gave you a very nice introduction. What do I want to say about? Well, just tell me. Well, I mean, I think since most of the audience is still young, no, and I assume some or many of you want to follow the academic career. I think it's always nice to see the research career, different research careers, because each of us has a completely different story to tell. So I'm an agronomical engineer from the Universidad Politécnica de Valencia. And then my first real research experience was during the bachelor's research project. But you know, I really liked it, but I wasn't very convinced about whether I wanted to follow the research career or not. And then I said, okay, I'm going to give it another chance. And then I got an internship, a fellowship, European fellowship, to go to Strasbourg, to the lab of Olivier Bonnet. And that is where I really fell in love with science. So I'm telling you this because even if you now go to do your master's thesis or even your PhD and you're not so convinced, I think it's worth, if you think you like research, just give it a try. So yeah, I definitely fell in love, and then as we went through, I did my PhD in Valencia, and then I moved to California, first to San Diego, where I studied a little bit of epigenetics and light signaling. And then I was lucky enough to go to Steve Jacobsen's lab, which as Juan Manuel pointed out, is probably one of the best labs in the world doing epigenetics in plants. And there I spent lots of time developing tools for epigenome engineering, which I will tell you a little bit at the end of the talk. But I think it fits really well in the common theme of this institute. And then in 2020, when I went back to Valencia, where I started my own group, and where we study epigenetics in plants. So yeah, this is the group. We're currently a small group. We've been there for 3 years, almost 4. We have four grad students, and then 1 postdoc, a couple of master theses, and then undergrads, and so on. So yeah, let's start with the science. So as you probably know, chromatin, which is the physiological form of DNA in the eukaryotic cell, plays a very, very important role in pretty much all the nuclear processes, yeah, like transcription, replication, repair, recombination, everything related to DNA. And it also plays a very important role in packaging the DNA, the very large molecule of DNA, into the tiny space of the nuclei. So the basic unit of chromatin is this particle here that is known as the nucleosome, yeah, that is formed by 4 pairs of histones, histone 3, histone 4, histone 2A, and histone 2B, and then it is wrapped by around 150 base pairs of DNA. So this is the first layer of compaction in the genome, and the position of the nucleosomes are very important, since the DNA that is associated to the nucleosome is not so accessible for DNA binding proteins. So for instance, if a transcription factor wants to access this region, but there is a nucleosome, it's going to have a hard time, and probably it won't bind to this region. So besides the function of the nucleosome, controlling the chromatin compaction and accessibility, it also plays a very important role in signaling, in nuclear signaling, because it is heavily modified with either histone variants as well as multiple post-translational modifications like methylation, acetylation, phosphorylation, ubiquitination, you name it. There are many, many. And all of these modifications can be recognized by specific readers, nuclear readers, that can identify a certain combination of epigenetic marks in the nucleosomes and then trigger downstream signaling events. So the distribution of these epigenetic marks and the compaction of the chromatin is not homogeneous along the genome. You can see all sorts of combinations that you can divide mainly in 2 parts. One that is the heterochromatin, where the chromatin is compact, there is no expression, and is enriched in specific epigenetic marks, like DNA methylation or certain histone modifications. And then on the other side, you have euchromatin, where the chromatin is much more open, is where most of transcription is happening, and it is also marked with specific epigenetic mark, like histone acetylation, for instance, is a classic euchromatic mark. So all of these modifications and the arrangement of the nucleosomes and the compaction and so on is the result of the activity of many nuclear proteins that can be divided in these families, either writers or erasers of these modifications, chaperones that help assembling the nucleosomes, for instance, readers, the guys I told you before that can recognize the different epigenetic marks, and then remodelers, which are our favorite proteins. Not only the distribution of the epigenetic marks is changes along the genome, but it also changes along the cell cycle, as well as during development and the response to environment. And this is true for all eukaryotes. Yeah? Epigenetic marks, chromatin is present in all eukaryotes, and it plays very important roles in all sorts of organisms. So in particular in plants, which is our favorite organism, well, there are many examples of changes of the epigenetic profile during the formation of leaves, flowers, seeds, etc., and also during the response to environment, which is a very important thing for plants, since they cannot move. Yeah, they're very good at responding and adjusting to the environment. One of the processes that is heavily controlled by epigenetics and chromatin is transcription. So here you see one example where you have a heterochromatic patch, nucleosomes are very close to each other, and this prevents the access of the polymerase, so you don't have transcription. And then on the other side you have euchromatin, which is more open, especially over this region that is the promoter. The promoter needs to be open in order to allow the polymerase to be recruited here and then start transcription. But think that even during transcription, the polymerase needs to deal with the nucleosomes. So where the nucleosomes are positioned is important for transcription. And I want to remind you one of the most important steps. I mean, I guess all steps are important during transcription, but one of the key steps is the initiation of transcription. So the selection of the transcription start site. Think about it. Who determines where a transcript starts? Which is the nucleotide where a transcript starts? As you probably know, genes can be transcribed from different positions. So for instance, this gene is transcribed from 2 transcription start sites that give rise to 2 different mRNA isoforms, no? This shorter isoform and then this longer isoform. And this is not trivial because the information that is present in the 5' of a transcript is very important for downstream events. Like, for instance, the translation efficiency of this transcript, the incorporation of additional amino acids to the protein that encodes, etc. So the selection of the TSS is very important, and it's not stable. It's actually quite controlled by the environment, by internal stimuli, so that you have different isoforms in different moments of a cell's life. Who controls this? Well, there is tons of research describing the function of the pre-initiation complex and the RNA polymerase II in the selection of the transcription start site. So this machinery, which has many proteins, basically binds to some 6 regulatory elements in the promoters of a gene, and then it positions the RNA polymerase II in the right position to start transcription. So this is quite clear. But chromatin itself, it also regulates the transcription start site. So there is research from yeast showing that the position of this nucleosome here, which is known as the +1 nucleosome, the first nucleosome downstream of the TSS. So the position of this nucleosome determines where the transcription starts. Although the mechanism in this case is not so clear. So as I mentioned before, the position of the nucleosomes is quite important and is very well controlled. So as I said, we have the +1 nucleosome downstream of the TSS, and then you have the +2, +3, +4, and the distance between them and the position is determined. So who determines the position of nucleosomes? So there are different factors. One of them is the DNA sequence itself. So it turns out that you can see here the DNA going around the histones, to form the nucleosome. So there are sequences that favor that the DNA can bend around the nucleosome. So this favors the formation of nucleosomes. And on the contrary, there are some other sequences like poly-A's, you see here, or poly-T's, poly-C, poly-G, that make the molecule more stiffer. And therefore, this prevents the formation of the nucleosome. So just the DNA sequence itself encodes the ability to form nucleosomes or not. And this has been shown in vitro and is very well known, but probably in vivo, the most important players are these guys here, the ATP-dependent chromatin remodelers. So this is a large family of complexes that use the energy from the hydrolysis of ATP to dissociate the interaction between DNA and histones. So DNA and histones interact, and then this machinery basically pulls from the DNA, then the nucleosome is looser, and then it can move along the DNA, or if it pushes very strong, you can even disassemble the whole nucleosome and then open the chromatin. So this is a summary of the different families. So you see there are many families, different subfamilies, and so on. They are all characterized by the sequence. They are all clustered by the sequence of the ATPase subunit, which is the one that uses the energy to move the DNA. But usually the ATPase works together with a bunch of other subunits. So you have the catalytic subunit, which is probably the most important. And then you have auxiliary subunits that help the complex go into the right places or do specific functions. So depending on the family, they can do different things. Some families can dissociate a little bit the interaction, and then they can push the nucleosome. So this is known as nucleosome sliding, and then they can open a little bit space so that the transcription factor, for instance, can access this region. Other can remove the nucleosome directly, so they make even more space. Or other families, for instance, participate in the incorporation of histone variants, like H2AC. Here you can see, so these remodelers are obviously conserved in pretty much all eukaryotes because they play very basic functions in the function of a nuclei. Of course, plants is no exception, so they're very well conserved. Here you see the domain architecture of the ATPases of different families, SWI, SWI, SNF, CHD, SWI1, etc. And not surprisingly, mutations in these ATPases have a very strong effect on the plant phenotypes. Yeah, they cause all sorts of pleiotropic phenotypes. This is general this is a general effect of epigenetic regulators. We were discussing this before, because you're messing up with all the nucleosomes in the genome. So of course you're affecting the expression of tons of genes, and then things go out of control. And these guys also have been shown to play a very important role in the integration of external or internal signals to transcription. So today we're going to be talking about one family in particular, which is the SWI SNF chromatin remodeler family. This family was first discovered in yeast many years ago, and most of the current knowledge we have comes from either yeast or animals. And the reason why there is a lot of research in animals, or one of the reasons, is because mutations in subunits of the SWI SNF complex are responsible of more than 25% of the cancers. 20-25% of the cancers. So think about it. So it has a lot of relevance. Obviously, there is a lot of interest and money on the study of the function of this complex. So they are the kind of complex that promotes sliding or eviction. So basically, the job of this complex is to open the chromatin to allow the transcription machinery and other things to access DNA. And one of the interesting features of this complex is that it can form subclasses. So the subclasses, well, this is like the general composition. So you have the catalytic subunits, and then, as I told you before, you have a bunch of other auxiliary subunits, scaffold, regulatory, and so on. But they can form different subclasses. So each of the subclasses is characterized by signature subunits. You can see them in different color, which provide the different subclasses with unique activities. Yeah, or unique targets, for instance. So this was very well known for animals and yeast when we started this project. It wasn't so clear for plants. So what's the scenario in plants? Plants have 3 ATPases of this family. They have Brahma, which is by far the best known. There are many, many studies showing how Brahma participates in the response to environment, development, many, many processes, even heterochromatin regulation and so on. Then we have Splate. There is a good number of papers as well. And then there is Minu, which at the time when we started, it wasn't so well known. There were like 2 or 3 papers about it, but the specific function wasn't very known for Minu. Yeah? But again, as I said, it wasn't clear whether plants had different subclasses of remodelers. So we thought, okay, let's take all the information that we know from mammals and from yeast, where the subclasses have been identified and the signature subunits have been identified, and let's take a look and see whether they are conserved in plants. So if a signature subunit from mammals is conserved in plants, perhaps plants also conserve the subclass. So long story short, this was a collaboration with the lab of Miguel Blázquez at the IBMCP. These are the results. A blue dot means the subunit is conserved. So you see all the subunits from mammals and from yeast, and all the different species we search. Representative species from metazoa, fungi, and many, many plants, from red algae all the way to Arabidopsis, dicots. So as you see, there are many subunits that are very well conserved. No, for instance, this is conserved in pretty much all eukaryotes. So this is telling you that this is a core subunit that is very important for the complex. However, there are others that are only present in animals, only present in yeast, only present in animals and yeast, but not plants. So this is the specifics of the different subclasses. So I won't tell you much about this. I want to focus on this guy here. This is a subunit that we identified present in plants. So until this study, this guy was thought to be only present in animals, but actually we realized that no, it is also conserved in yeast and in plants. I guess many people never look in plants. No, it's one of the, like animals and yeast, they do their own thing, but they forget about the plant kingdom. But yeah, it's very well conserved in plants. And we got interested, of course, in it. What was the function of this protein in plants? So we name it triple PhD fingers. And the name is obvious. It has 3 PhD domains plus 1 two-door domain. So both PhD and two-door domains are histone reader domains. Yeah, these 2 domains. So as you can imagine, this protein is basically a histone reader that is incorporated in SWI SNF complexes to help going to specific regions of the genome. So the first thing we did was to validate that indeed this protein that we identified in an in silico assay, yeah, the phylogenomics assay, is actually a component of SWI SNF. So we made Arabidopsis transgenic plants expressing TPF1, this guy here, fused to a tag, 3x flag. And then we did immunoprecipitations followed by mass spectrometry to identify the interactors of TPF1. And indeed, what we found is that TPF1 pulls a whole SWI SNF complex. So this makes a lot of sense. But this complex has some interesting features. Yeah? One of them is that it incorporates MINU as the ATPase. See, if you remember before, I told you plants have 3 ATPases. So this complex incorporates only MINU, which is already telling us that perhaps there is a subclass in plants that all incorporates MINU and it has its own function. But moreover, this complex incorporates 5 other plant-specific subunits that are not present in the complexes bound by Brahma or by Splate. So this was the confirmation that plants have different subclasses, and this is one of them. And moreover, plants have incorporated these 5 additional subunits that are not present in animals or yeast. So perhaps they're doing something special, or they adjusted to the plant biology. I don't know. We're studying right now the function of these guys. And well, we named the complex the mass complex for MINU-associated SWI SNF complex. And as I told you, it has a few signature subunits. TPF is one of them, and then the 5 plant-specific signature subunits. And yeah, we started to characterize the molecular function of this complex. Nobody had done it before. So we did a bunch of experiments. We first characterized the mutants of TPF, the subunit we found and that we used for the mass spec. And then we did a bunch of genomics experiments to characterize what the function of the complex. I will tell you about it. So for the TPF mutant, first thing we did, we ordered tDNA mutants, which, as you probably know, are mutants that you can find in public repositories, where a tDNA is inserted in the gene, so it disrupts the function. Here you can see where the tDNAs are inserted. So we got a couple of alleles for TPF1, a couple of alleles for TPF2. As you see, there is no phenotype. This is a wild-type plant. These are the mutants. So. But then when you cross TPF1, TPF2, then you have a very, very strong developmental phenotype, which is telling you that TPF1, TPF2 are probably functionally redundant, which is something quite common. And then depending on the allele combination, you will get stronger or weaker phenotypes. So we had alleles of different strength. Fine. So this is a summary of the phenotypes we characterize for TPF1. Here you can see that the strong TPF1 mutant is smaller compared to wild type, has shorter roots, is developmentally delayed. The meristem is smaller. You can see it here very well, but it has problems with the plane of division. There is a whole pool of cells that are dying in the meristem. Perhaps we can talk about it later. And yeah, these are the adult plants. As you see, this is wild type, this is the mutant. The plants are dwarf. They produce many, many lateral branches. And one important thing is that the TPF mutant resembles a lot a MINU mutant. MINU is the ATPase of the complex, and TPF is TPF. So because the phenotypes are very similar, this is already telling us that TPF function must be very important in the complex activity because it resembles an ATPase mutant. And then, well, there are all sorts of problems with the flowers, with abnormal organs and so on, and then it ends up producing these tiny silics that are empty, so this mutant is sterile. So yeah, TPFs are very important for development. Then we went and dive into the genomics to characterize the complex, and the first thing we did was to figure out where is the complex going to, which are the genomic targets of the complex, and for this we did ChIP-seq. So I'm sure you're all familiar with what a ChIP-seq is, but very briefly, You get the chromatin of your favorite mutant or plant, and then you fix the interaction between the proteins and the DNA with formaldehyde. Then you cut the chromatin with sonication, and then you immunopurify your favorite protein using antibodies. So because your protein is associated to DNA, you're immunopurifying your protein. Then you recover the DNA that is bound to your protein of interest, and then you make libraries, sequence, and map where the DNA where the DNA comes from, and then you can infer where your protein of interest was located. And yeah, just as a little reminder, then you get these sort of representations where you have peaks, and the peaks are the result of the summary of lots of short sequencing reads. Sorry. So see, here you have a peak, here you don't have a peak because you don't have this pool of sequences. So these are the peaks that we see for TPF1 or TPF2 or MINU2. I forgot to mention, but MINU1, Arabidopsis has two paralogs of MINU, MINU1, MINU2. So we characterize MINU2. As expected for 3 proteins that belong to the same complex, the peaks overlap very well, as you see here. And if you do a metaplot representation, you see that all 3 proteins, so the complex, is binding right downstream of the transcription start site. This is the transcription start site, this is the termination site, promoter, terminator. So it peaks right downstream of the transcription start site. Yeah? And the localization of the complex, so here in these heat maps you can see where TPF1 is bound. These are 16,000 genes. TPF1, TPF2, MINU, they colocalize nicely. And they colocalize nicely with a bunch of euchromatic marks. Acetylation, H3K4 trimethylation, H3K36, these are classic euchromatic marks, so promoting expression. And it anti-correlates 100% with heterochromatic marks, like K27 trimethylation. See, where the K27 trimethylation there is, there's nothing, no complex. So this complex is sitting right on the transcription start site of expressed genes. This is what we know up to now. Of course, this is a chromatin remodeler, so it's obvious to go and look for the nucleosomes. What's the impact of the mutants of MINU or TPF on the nucleosomes? And for this, we did a MNA-seq. So I did this little cartoon for you to see what the MNA-seq is. You basically get your chromatin, and then you digest it with micrococcal nuclease, which is an endoexon nuclease that degrades much faster the DNA not associated to the nucleosomes than the DNA associated to. So you basically digest your chromatin. This guy is going to quickly digest the open chromatin, the open DNA, and it will preserve these guys, which is the DNA associated to nucleosomes. Then you purify this DNA, make libraries, sequence, map it against the genome, and then you will get this sort of mountains that will tell you where originally the nucleosomes were. And this is how MNA-seq tracks look like. So you have these mountains. See if this is the +1 nucleosome right downstream of the transcription start site, +2, +3, +4, and so on. So when you're doing a MNA-seq experiment and you're comparing a wild type and a mutant, obviously you're looking for differences, and these are the 3 kinds of differences you can expect. You can expect that in your mutant or your condition, the nucleosome moves. So that's a position shift. There could be a change in occupancy, meaning that there is less nucleosome or more nucleosome in your mutant, or you can have changes in the fuzziness, which represents the position of the nucleosomes across thousands of genes. So if all the different cells have the nucleosome position in exactly the same position, you will have a very sharp, nice peak. If each cell has the nucleosome slightly changed, you do the average, you will have a lower peak, more like broader. Anyway, so we calculated which kind of changes there were in the TPF or in the MINU mutants, and as you see, most of the changes were related to changes in position of the nucleosomes, which is this example here. So this is one example where you can see it clearly. This is +1, this is the +2. So imagine these are the nucleosomes, and then both in TPF or MINU, which are the same mutant pretty much, you see there is a small shift towards the promoter. It's small, but it's very, very consistent. So this will be represented by this move. And the move is going always towards the promoter. Yeah? So this is true not for one gene. This is true for thousands of genes in the mutants. So we do this sort of representation where you see the nucleosome positioning. This is the +1 in wild type over thousands of genes. +2, +3, +4. Then this is how it looks in TPF and MINU. It's impossible to see anything here. But if you do the difference TPF minus Columbia or MINU minus Columbia, then you see these plots where red means that the nucleosomes are moving towards the promoter. So the +1 nucleosome moves over thousands of genes, the +2, the +3, the +4. And the phenotype is identical for TPF or MINU. So this is something general for thousands of genes. Then you can represent this data in a meta plot. So it's same. This is the +1, +2, +3. In gray, you have the wild type. And then in orange and blue, you have the mutants. So it's a small shift. And we can quantify it. In average, it's 10 to 15 base pairs. So we don't know if this is functionally relevant or not, that a nucleosome moves 10 to 15 base pairs. But indeed, this data is an average of thousands of genes. But there are hundreds of genes where the nucleosome moves 50 base pairs, even 100 base pairs. So for now, we have that the complex sits downstream of the transcription start site of expressed genes, number one. Number two, we know that in the mutant, the nucleosomes move to the promoter. Next question is, okay, the nucleosomes move, what is the impact on a gene expression? Has it any relevance at all? And then, of course, we did RNA-seq. So we did RNA-seq of TPF or MINU mutants compared to wild type. Not surprisingly, we see thousands of genes that are expressed upregulated or downregulated because the plants have pleiotropic phenotypes, so you would expect this. But these misregulated genes could be a direct consequence of the activity of the complex, or it could be just a secondary consequence, because these primary targets are misregulated and then they misregulate secondaries, which are misregulated. So we focused on the direct targets, which are the genes bound by the complex and misregulated. So there are 427 upregulated genes and 500 downregulated genes that are usually bound by the complex. So the first thing you have to realize is that most of the targets of the complex don't change the expression. So this complex binds to 16,000 genes. 15,000 of them, once you lose the complex, they don't change. So what is the complex doing there? But we thought, well, perhaps those guys that become upregulated or downregulated are the genes that show a lot of change in the nucleosomes or something. So we look for this. We look for the direct upregulated, direct downregulated, or direct non-differentially expressed. How many of those the nucleosomes move? This is the blue bar. For all three sets, the nucleosomes move in around 1/3. So there is no connection. No matters if the nucleosome shift, that has no effect because it moves the same in direct up, direct down, or even direct that are not differentially expressed. So what is the effect of this nucleosome change? Maybe it has no effect. And we were lost for a little bit, but then taking a look carefully to the data, to the tracks, this is one of the examples I show you, nucleosome shift. Yeah, you see the +1 here, peaks right here, and then little shift towards the promoter. And then we realized, looking at the RNA-seq tracks, that many had this phenotype. So take a look to this. These are the RNA-seq reads for this gene in wild type. So they accumulate to the first exon and then the second exon. And then look in the mutant, these reads are the same, but you have this additional accumulation of reads towards the promoter. And then we found out that there is a very strong connection. So every time we would see this case, there was a change in the transcription start usage, because this is at the end the result of starting transcription from upstream position. And this is, we see it over thousands of genes again. So there is a very, very strong correlation between genes that change the nucleosome position, which are these guys, and genes that start the transcription towards 5'. So there's a functional connection between the nucleosome position and the transcription start site. Next question, okay. These reads, where do they come from? No, perhaps these are small reads that the RNA polymerase is transcribing, but that the RNA polymerase stops transcribing, and there are short fragments that map here, or they could belong to full-length mRNAs. So we did this silly experiment where we did a cDNA, qRT-PCR, designing oligos that go to the end of the gene and to the beginning of the gene, but mapping to the longer isoform. And indeed, this is what we see. The mutants produce with much more frequency this full-length mRNA isoform. So we conclude that these are not short transcripts. These belong to long mRNA transcripts. Okay, so this is one story, and then in the next 5 minutes, I would like to tell you very briefly about what I did in the postdoc. Because I was yesterday looking at the web page, and then I realized this is the Bioengineering Institute, and I was like, okay, wait a second, I've been doing this for like 6 years, so I should say something about it. So during my postdoc, as I briefly mentioned before, in the Jacobson lab, I spent lots of time doing epigenome engineering, which is a very cool way of doing bioengineering, where basically we developed tools to manipulate the epigenome in a targeted manner. So yeah, as we discussed before, there are many different epigenetic marks on the DNA and the histones. The question is, can you manipulate the epigenome in a controlled manner? The answer is yes. You can do it with more brute force, like you can just overexpress epigenetic regulators. You can express your DNA methyltransferase or your histone demethylase or whatever, and then it will modify all the genome, and then you can go and do data mining and see, try to correlate changes. A better way of doing this, in my opinion, is doing targeted manipulation. So basically, you would target your favorite epigenetic regulator to one specific region in the genome, and then you would explore the direct impact of this manipulation on one specific gene. You're not dealing with like genome-wide changes. Why is this important? Well, first of all, because it allows you to study epigenetic regulation. And this is very important because when we use these mutants, and I just show you one example, since these mutants affect the whole genome, you have pleiotropic phenotypes. And it's very, very hard to figure out what is direct effect of your regulator versus indirect things, because it's all mixed. You just have a stable mutant. With this way, you can, with very high precision, manipulate the epigenetic mark of one gene at one specific position and study exactly what is going on. So much more precision and direct consequences. And then second point of interest, you can generate new epialleles. And this is very, very interesting for applied research. Like, for instance, breeders, they're always looking for genetic variability, no? And in this case, you would generate epigenetic variability that they could use for breeding programs to just increase their tools to improve crops and so on. So how do we do this targeted manipulation? We use programmable DNA binding platforms. What is this? These are proteins like artificial zinc fingers, style effectors, CRISPR, that basically we know very well how they bind to DNA, and we know how to reprogram them to change their targets. So we can reprogram the zinc finger, and instead of going to its natural target, it will go to our favorite gene, to the promoter or to the terminator, whatever. Same for style effectors, same for CRISPR. And then once you've reprogrammed these guys, you can fuse it to whatever you want. You can fuse it to a GFP, if you want to see the fluorescence of your favorite gene. You can fuse it to histone modifiers, activators, repressors, DNA methylation machinery, which is what I did during my postdoc. And, well, very long story short, what we did is we used artificial zinc fingers. We fused them to DNA methyltransferases or DNA demethylases to manipulate DNA methylation levels over specific locus. And we also used CRISPR. I'm sure you're all familiar with the CRISPR system. So CRISPR, you have two components, yeah? You have the guide RNA that gives the specificity of target, then you have Cas9. Well, in this case, you don't want active Cas9, you don't want the Cas9 to carry it, you just want the Cas9 as a bus that is bringing your effector, so you have to use deactivated Cas9 that will sit on your target, and then you can fuse whatever you want. In our case, we use the CRISPR SANTAG system. So this is a very cool improvement of the system where you add an additional C-terminal tail to Cas9. And this C-terminal tail has 10 copies of an epitope called GCN4. The name is not important. So this, on top of expressing this modified Cas9, you express a second module that has a single chain antibody that can bind to the GCN4 epitope, fused to whatever you want. In our case, DNA methyltransferases or demethylases. So the difference is that with just one Cas9 molecule that goes on the target, you can recruit 10 effectors, while when using zinc fingers, you can recruit just one effector. So this is way more potent and specific. This is very, very specific. We see that once you use this, it just goes to one gene. Zinc fingers are more leaky. They tend to bind to more places, but this is like a very powerful tool. And we show, that with these tools you can manipulate in a locus-specific manner the DNA methylation levels. And the cool thing is that we even got stable, heritable, transgenerationally heritable epialleles that maintain the changes in gene expression. Indeed, these sort of tools have been used recently in cassava by the group of Becky Bart in the Danforth Center to manipulate, as I said, cassava to change the response to a pathogen. So this is actually something that is being used currently to improve and to solve real crop problems. It's not just like basic science. And these are some works that we studied that you're very welcome to take a look if you're interested. And this is it. Just to thank the members from my group. Pretty much everybody has participated here. Collaborators from the IBMCP, Spain, US, France, of course, funding agencies, and thanks a lot for listening, and I'm happy to take any questions.